Welcome back to McGill's first bicentennial cutting edge lecture in science. Introducing our speaker is the student ambassador, Adeline Lee.
So hi everyone, today we have the first talk in the series titled Unraveling the Secrets of Symbiosis, How Ants and Bacteria Became One, which will be presented by Professor Ehab Abouheif from the McGill Department of Biology. Now, today's talk will be, I guarantee, full of twists and turns, and like many discoveries in science, the story we will be hearing today actually began as a total accident. So how did all this happen? Let's look forward to the talk then. Well, depending on how much time we will have at the end, we may have a Q&A session. So it's important to stay tuned after the talk. So without further ado, Professor Abouheif, I'll hand it over to you.
Okay, this is a real honor to be able to kick off the series of the bicentennial lectures. I hope to share with you about a field called Evo-Devo, or evolutionary developmental biology, as you can see over here. And I want to share with you our Evo-Devo journey to understanding the secrets of symbiosis. And I hope you just hang on to your seats as we go on for this ride. Okay, and so really what we're trying to understand with the field of Evo-Devo is how this remarkable diversity of nature, as you can see here in the slugs, to sea stars, to crabs, to insects, how do all these marvelous forms in nature evolve, okay? And this is really one of the primary goals of Evo-Devo. Now, one of the major discoveries of Evo-Devo, it's actually a very old field, but over the last 40 years, this field has made remarkable discoveries that I'd like to share with you today. Essentially, what we've revealed is that all these remarkable forms here are regulated by the same genes, what we call a highly conserved genetic toolkit. So all of these animals, from jellyfish all the way to humans, have the same genes that regulate development. And let me share with you just a little bit about what this genetic toolkit looks like. And so here is a gene that we call engrailed. And what you're looking at here in green is the embryo of a fruit fly. The head is over here to the left, going all the way around, spinning. What you can see in green is the expression of this gene engrailed, marking where the future segments of the fruit fly in the adult fruit fly are going to form. If you zoom in on these segments, you can see here, they actually mark, you see the cells of where these segments and segment boundaries are going to form. And these genes are highly conserved in evolution. So if you look at this gene engrailed, whether you're looking at this fruit fly, the same embryo but filleted open over here on this side, you can see the reiterated stripes where the future segments are going to form. Whether you're looking at a fly, a beetle, a bug, a crustacean, a worm, or even a fish, or even a human, you will see the same patterns of expression of this gene playing a role in setting up future segments. Now, humans, we don't have visible segments on the outside, but our muscles are segmented, and that's where you see the expression of this gene engrailed. Another remarkable discovery in Evo, called the Hollywood experiment. Here you have different eye types. So this is the eye of a fly, the eye of a human, the eye of a cephalopod, and the eyes of these are the remarkable eyes of flies, set up with all these little tiny little visual systems, which looks very different to the camera eyes of vertebrates or even that of a cephalopod. At the same time, we think of these as structures that evolved completely independently of one another. They're not really related. But what was covered by Walter Gehring's group in Switzerland is that the gene that regulates these different eyes during development are highly conserved. And in fact, if you take the gene from a mouse and overexpress it during the development of a wing of a fruit fly, you can get an eye. This is a mouse eye gene that you express in a fruit fly, and you can get the eye to grow on the wing of a fruit fly. And in fact, if you express it anywhere, if you express it during the development of the head, you can get eyes on the head, wings, eyes on the antenna, eyes on the legs, eyes on the antenna. And so what's happening is you don't get a mouse eye, but the mouse gene is able to activate the cascade of the fruit fly. Another set of remarkable genes that the field of Evo Devo has discovered are what are called Hox genes. Now, these Hox genes are absolutely fascinating and remarkable. Essentially, these genes tell the identity along the anterior and posterior axis of the animal, where exactly what the different body parts will be. And if you look along the chromosome where the Hox genes are situated, they're aligned, they're basically aligned on the chromosome in the same order that they're expressed along the body. And so the gene called labial here in red is expressed in the head. And as you go down, antennapedia here, which we'll keep going, is expressed in the second thoracic segment, UBX in the third leg and third segment, and so on and so forth. And so this is how they're expressed in a linear order across the chromosome. And so they tell basically, if you take on its anterior posterior axis, says, okay, this part of the body at the top is going to be the head. This is going to be a leg. You're going to grow a leg here. You're going to grow, a wing here, and so on and so forth. And so if you look at the expression of these genes, again, along a fruit fly embryo, this time it's expressing all these genes at the same time. If you mess with these genes, if you somehow, we have the ability to knock down these genes. So if you knock down a gene called antennapedia over here, something remarkable happens, okay? What happens is you can transform, this is a normal fruit fly head. And what you see here, these two structures, these are antennae. If you basically knock down the expression of antennapedia, you can get legs growing out where the antennae are. And so you're basically transforming the identity of different parts of the body by messing with these Hox genes. What about the gene UBX? If we knock down this gene UBX, essentially, if this is a normal looking fruit fly over here on the top with a pair of wings, one pair of wings over here, these are balancing organs called halteres. If you knock UBX down, which make these halteres, you can get a four-winged fly. Okay, and so we start to mess around with basically the postal address or the identity of where the body parts are supposed to form. And of course, these Hox genes, like all the other genes in this genetic toolkit, are highly conserved from, again, jellyfish to humans. And so here you can see the Hox genes arrayed in the same expression in a fruit fly. And this embryo, you can see that the mouse also have these Hox genes are expressed in the same order. And so what we think, of course, basically the status quo, or what we think about in the field, is that this remarkable diversity of forms is basically comes about by tinkering with the number and the timing of expression or the regulation of these genes that I'm telling you about, whether it's engrailed or it's PAX6 or these Hox genes. This is how all this diversity comes along. There's one basically discovery by Conrad Waddington that is really quite remarkable and basically set up my whole program of research. And that is these genes can respond or basically can respond to temperature and different environmental conditions. So temperature or cold shocks, if you expose a fruit fly embryo to a temperature or cold shock, you can mess up with this gene UBX and you can produce a four-winged fly. This means these genes, these genes are not commanders. They don't tell basically this is how it's going to go. And if you tinker with the number, it's not only tinkering with the number of regulation, but this genetic toolkit interacts in a very complex manner with its environment. Like I just showed you, if temperature affects this this gene UBX, you can get this four-winged fly. And so it makes this really complex interaction with genes and the environment to kind of form how organisms basically develop and evolve. And that's what I've spent the last 20 years of my life trying to figure out. And the way I've been doing that is looking at the remarkable diversity of ants on the planet. Now, of course, all of us have seen ants in our kitchen and different, just walking along the ground, but you may not realize how complex their societies are. There are essentially 20,000 species of ants, in the world. And if you were to take all the ants in the planet and all the humans on the planet, they would form equivalent blobs, according to Dobler and E.O. Wilson, two very famous ant biologists. Now, you can even just see here, this is looking at the fronts of the heads of ants. You can see they all belong to a single family, but even though you see a remarkable diversity of forms. I never knew that if by looking at it, you would see an ant that looks like Spider-Man or either one that might look like Darth Vader. Now, this whole story started because we were trying to answer a fascinating question in terms of body plans. And it turns out, that ants are so diverse and so prominent on the globe that there are many insects that live around the ants that want to mimic the ants. They want to look like the ants. And so what's remarkable is that you have many different insects, whether it's a cricket or a spider or different kinds of things that actually they've evolved to look exactly like the ant body plan. And one of the remarkable features here, this is a bug, this is a cricket, this is a spider. And one of the remarkable features of the ants is that they have this constriction called the petiole to understand this process. And so we started to turn to looking at this wonderful group of ants called the Carpenter Ants. And these Carpenter Ants are hugely successful. The number of species in this single genus of ants is far over 1,000. That means they make up about 7% of all ants. And they're far more diverse than all the other genera that exist in the ants. Now, to try and understand this, mimicking of body plans of insects, mimicking plans of ants, I had a postdoc in the lab by the name of Dr. Mateen Rafiki. And he was starting to try to understand this. And by trying to, the first step was we use these Carpenter ants just to look at this genetic toolkit, the Hox genes. And so what he did is this is an embryo of the Carpenter ant, a late stage embryo of the Carpenter ant. And he looked at the expression of this Hox gene, UBX, and this is the same one that gives the four-winged fly. And what we thought is that, okay, well, if we mess in purple with this purple expression of the, change the identity of body parts, we might make that constriction turn into another different body part. But something really odd happened here. And instead of changing the expression of body parts, we ended up completely truncating the embryo. This is what a normal sort of late stage embryo of the Carpenter ant looks like. Instead, we almost cut it in half. So we just had the head and the first three segments, and this is where the expression of the gene stops. We cut off that whole part of the abdomen. Here it is. This is kind of, you can see that we completely, knocked it down, truncated it. And so we really thought, Mateen and I, we had intense discussions about this, and we thought we had failed. We thought the experiment didn't work. We thought this was never been seen before. Usually, like I said, you mess with the postal address of these Hox genes, you change an antennae where you put legs where the antennae should grow, you put wings where wings don't normally grow, but you never expect to completely chop in half the embryo. So we thought we had failed, and we put the project aside, ready to give up. Meanwhile, we had two undergrads in the lab, Travis Chen and Ben Feng, who had joined the lab just to do some really exciting undergraduate research at McGill. And they did some from the Florida Carpenter ant. Again, this is a queen that's sitting in the middle. These are the eggs that are surrounding, and these are the workers that basically are all around taking care of the eggs. And Ben and Travis got really excited and wanted to do all kinds of things. But what we decided to do was make a developmental table of the ants. Like what does an embryo look like after 2 hours? What does it look like after 4 hours? What does it look like after 10 hours? And so on and so forth. And so they did this remarkable work where they, we had different stages of the Florida carpenter ant. And they made this poster for an undergraduate symposium, and we put it up on the poster in the hallway. And one thing that we kept arguing about, essentially, is this is the first stage 1 egg of the Florida carpenter ant, and there was this white cloud that we saw in the posterior part of the egg, and this is not something we've ever seen in any other insect. And we're like, what is that cloud? This is really bizarre. And I guess since Mateen had failed his project, we kept arguing. Sometimes we thought it was, mRNA. Sometimes we thought it was DNA. Sometimes we saw some kind of protein. Sometimes we didn't really know what it was. And so Mateen's project thought it had failed. And Junie, a senior PhD, now a student in my lab, who's going to be starting his postdoc soon at MIT, and Mateen is now a professor in Istanbul, Turkey. They got together and they started to try and understand, well, what is this cloud doing? And these gentlemen are incredibly talented, did some remarkable work. So this is what this cloud would look like, as you see. And so they started to do some basic experiments together with Travis and Ben. And we said, feed it with DNA, and DNA is an enzyme that eats up DNA. When they did that, the cloud had disappeared. Wow, that's great. When we treated it with RNAs, an enzyme that eats up at RNA, well, the cloud remained. And when we treat it with xylenes that eats up protein, it still stayed there. And so whatever this cloud is, it had to be composed of DNA. And so we thought we had discovered this remarkable new mechanism where the mother is dumping her DNA into the, and this would be a completely novel way of transmitting DNA from one generation to the next. And we presented a poster as such in Paris. But while we were in Paris, there were some researchers that saw this and said, you know what, I don't know if this is DNA. We've seen that cloud before, okay? And when we looked at this cloud, we took a closer look. We listened to them. We took a closer look. And what we discovered was something remarkable, that it wasn't DNA that the mother was done, it was actually bacteria. And so you can see here the strands of the bacteria in that posterior part of the egg. And here, if you squish the egg, you can see the bacteria that are swimming along in that posterior part of the egg, okay? And this bacteria is called Blockmania, okay? Now, once we knew it was bacteria, we started to look around the literature. And what we realized is that a gentleman by the name of Blockman had discovered this bacteria in 1884. And we felt, quite, astounded that this level of research, if you see the drawing that Blockman had done, the level of detail, it's almost as close to our modern microscopes that you see today. Now, to be fair, Blockman himself in 1884 thought that this was a fungus. He didn't actually get it. It was only 20 years or 30 years after his discovery that they realized that this was actually a bacteria. So it didn't make us feel too bad, but it just goes to show you that over the course of 150 to research is forgotten and we turn our wheels. And this is the nature of discovery, that we forget the past and we move on to the future. And I think it would be wise to reflect on the past as well as the future when we move forward. Now, what's amazing is we started to get a hold of what this bacteria is doing. This is an ovary of an ant, of the carpenter ant that we're showing you. And here the bacteria is in white. The bacteria comes in and starts to infect the ovary about halfway through what we call oogenesis. Then the bacteria get dumped into the oocyte, with this future egg, once the queen lays it. Then the bacteria starts to localize slowly and surely to the posterior pole, and it surrounds this yellow circle, which is the germline. This is the future genes. This is the genes that get passed on from one generation to the next. And it completely surrounds the germline. Now, what we didn't know is we wanted to know, well, what happens, now that we knew what was happening to these carpenter ants, we realized that this bacteria actually plays an incredibly important role in the life of these carpenter ants. For example, the carpenter ants, the reason they call them carpenter ants is that they have colonized a completely new environment, which is the wood. Now, carpenter ants aren't termites. They don't eat the wood. They actually live inside the wood. And we think that this bacteria is critical for allowing them to live in the wood by enhancing their immunity, allowing enhanced nutrition and different kinds of things. And it's remarkable. These are carpenter ant workers. This is a larvae of a carpenter ant. If you split this, it almost looks, it's quite remarkable. This is what a carpenter ant larva looks like. And if you look, this is its gut. So just like humans, ants have bacteria in their guts. And if you look at it, you can see the bacteria are swimming all around their guts. Now let's take a closer look. If you take a closer look at that, you can see the bacteria that are basically, each one of these little circles is a cell. And each cell, which are these cells are called sites because they harbor the bacteria. And each of them is packed with bacteria inside the gut of the ants. Now let's take a closer look. If you take a closer look at each one of those bacteriocytes, it's absolutely mind-boggling. Each single cell is jam-packed with these bacteria. They almost look like strands of spaghetti. And it's remarkable that this cell can even function. This is the nucleus of the cell. This is the membrane of the cell around, and this is the bacteria that are jam-packed in each of these cells in the gut of the bacteria. Now, when I first gave this presentation to my colleagues, my scientific colleagues, it was great because on Twitter, this was one of my favorite tweets that came out after my talk. And basically it said, who wore it better? So this is one of my favorite tweets of the thing. So what does this do? I mean, what we realized from looking at the literature and this amazing research by my colleagues in the field, essentially showed that this Blockmania bacteria can't live outside of the ant. It dies. And the ant without this bacteria, it will die. It's what's called an obligate endosymbiosis, that their lives, they're a single, almost like a single individual, that their lives are inextricably linked. Now, the Blockmania provides enhanced nutrition and immunity to the ant, and the ant in turn provides to it this bacteria, it gives it a protected cellular environment, and it makes sure that it gets passed on from generation to generation. So this is basically a mutual, obligate symbiotic relationship that they must live together. Now, if you lose Blockmania, I just wanted to give you can treat these with antibiotics, and colleagues have done this, and they've showed that if you lose the Blockmania, not only does the colony start to disintegrate and fall apart, but you take this whole range, these are all the different kinds of workers in the carpenter ant colony, you almost have the size of the workers when this happens. And so this is really, a necessary part. They can't live without each other. Okay. So let's step back. Now we know that this remarkable symbiosis that we see in the guts of ants through this Blockmania bacteria is not just an ant thing. Symbioses are in all of life. And I just want to show you how symbiosis has essentially changed our picture of life over, ever since Darwin published his Origin of Species in 1859. Now, of the, many hundred pages in the Origin of Species, he had one single figure in the Origin of Species. And what you see here is a branching diagram, a tree of life. He drew a tree of life that goes back to our ancestors, and splitting over successive generations, these they split and evolve and change. But at that point in 1859, we saw this splitting as going from the ancestors to it was basically a very binary tree that goes from ancestor to descendant. many years later, that sometimes, rarely, on rare occasions, symbioses happen. And this is work, sort of these cartoons come from the remarkable work of Ford Doolittle, and of course, Lynn Margulis, who came up with what's called the Endosymbiotic Theory, the idea that our cells today, the eukaryotic cell that gives rise to the human body in animals, plants, and fungi, these were the product of two different unicellular bacteria that symbiosed together and gave rise to the cells that we know in all animals, fungi, and plants. But it was thought to occur very rarely in the tree of life. It wasn't something that really challenged sort of our ideas of the tree of life. And what later, what we've come to appreciate, this is another drawing by Ford Doolittle, is in fact, symbioses are a lot more common than we might expect. And instead of thinking of a tree as going from ancestor to descendant, now we realize that symbioses are everywhere and crisscross in remarkable ways, messing up our version of a clean version of a tree of life. The tree of life is remarkable in its symbiosis as it changes our view. Let me give you a few examples that this is not, that what we're trying to understand here, the way that bacteria and ants is actually something that happens in all of life. This is one of the most mind-blowing kind of examples of symbiosis that occurred that I learned about from my colleague Brian Hall and Ryan Kearney at the University, Dalhousie University in Halifax. This is the spotted salamander. And this spotted salamander evolved a symbiosis with photosynthetic algae. You can see the little eggs of the salamander completely by algae. The waste products of the salamander is good for the algae to grow, and the algae gives then enhanced sort of energy to the salamander. This also happens, this symbiosis with photosynthetic algae also happen in sea slugs. And so you can see how this tree of life is a merger between bacteria, algae, fungi, and the animals, in this remarkable symbiosis that happens in life. Well, this shouldn't surprise us, of course. Humans, the human body contains trillions of microorganisms, which outnumber human cells about 10 to 1, okay? And the microbes affect our digestion, immunity, disease, mood, and even possible behavior. A book that I would recommend is by Ed Yong. It's called I Contain Multitudes, that discusses this remarkable symbiosis between bacteria and humans. And it's called essentially the Human Microbiome. And really challenges our notions of what is an individual. If we have bacteria that we cannot live without, that outnumber our own cells by 10 to, about 10 times, what does it mean to be an individual? This is quite remarkable. And it's been proposed that essentially, like, the merger between these symbionts, bacteria and the ants, algae and the sea slugs, algae and the spotted salamanders, the human and bacteria, are called the holobiont, that we form essentially a single individual. It's quite a controversial topic, but it's really thought-proof that makes us think about what it means to be individuals. And these of course, these symbioses are absolutely key for understanding health, disease, our own particular state. Okay, so what we don't know, what really is unknown, we wanted to understand. All these remarkable symbioses have been documented. People have done exquisite research on the function of certain bacteria in the human microbiome. But what we didn't understand, nobody still understands, how does it happen? I mean, just think about this. How does it happen that two completely unrelated organisms, like an ant and a bacteria, how is it that they originate and evolve this symbiotic relationship to the degree that they can no longer live without each other and have to, basically that they merge? And so nobody really knows this. And so together with Mateen and Juni, these really remarkable scientists, we put our brains together and figure out what are in fact the steps by which Blockmania and the ant, this bacteria, Blockmania, and the ant came together. And so we started to investigate, and we started to take, the literature and started to piece it together. What we knew is that this obligate endosymbiosis between ants and bacteria happened about 51 million years ago. And one of the clues that helped us understand what was happening is, the theory is that it essentially came from bugs, or these bugs, that ants form an ecological mutualism with these bugs. These bugs, these are unrelated insects. The bugs essentially, they have a little basically beak or rostrum that they put into the plant. They suck up the juice of the plant. They have bacteria in their guts that help them digest, the juices from the plants. And then what they do is they exude it from their butts. The ants in turn drink this and they get nourished. And what the ants do for these insects is they then protect the insects in return, and they form this mutualism. And the theory is that, of course, this is how the ancestors of Blockmania passed into the ants 51 million years ago. And so our question was simple. When Blockmania came into Camponotus, did it influence its germplasm? As I showed you before, the bacteria completely surround the genes that are going to be the germline, which is going to be passed on from one generation to the next. And so we started off, again, we looked into this Evo-Devo genetic toolkit and looked at these genes. This is a gene called Oscar nanos, which is highly conserved. Basically, they're expressed at the posterior end of the embryo, either in fruit flies or wasps or in other kinds of insects, and they form the germ cells. And again, these are the cells that are going to be passed on from generation to generation. They're kind of like forming the sperm and eggs that we will pass on from generation to generation. And you can see that these genes, what we call germline genes, are expressed in a single location at the end, okay, of the embryo, and they will then form the germ cells that will get passed on to the generation. Now, these germ cells, these genes, have two roles. Not only do they form the germ cells, you can see here, this is where, just to kind of give you an idea, this is where we're talking about. The mother dumps in these genes, they get carried to the end, and they form the germ cells here. This then acts a whole set of genes that form the segments. And then once the segments are formed, this is like the gene engrailed that I showed you. It's all over the embryo in stripes. Then you get the Hox genes that basically tell the address of each segment and what it's going to develop into. So the Hox genes only turn on at the very end, but these genes get turned on at the very beginning, tell the embryo which is the head, which is the tail, and form the germ cells at the beginning. And if you mess with these genes, not only do you, like if you knock down these genes, not only do you basically get rid of the germplasm, so you get rid of the hereditary material that's going to be passed on from generation to generation, but you eliminate the whole posterior half of the embryo. So it tells these genes tell the embryo where the head, where the tail is going to be. So these are telling them, this is your tail. And if you get rid of them, the tail is going to be completely truncated, and it's going to form those germ cells that get passed on from generation to generation. So what we started this research, Mateen and Juni, essentially looked at what they call the common garden ant, Lasius, which is a cousin of the carpenter ant. Okay. Now this cousin, the common garden ant, we see everywhere around the world, and it doesn't have this symbiosis with the Blockmania bacteria. And if you look at these germline genes that form the germ cells and tell the embryo what's head and tails, the mother, like I said, dumps in these genes. These germline genes get carried to the posterior pole, and they localize to one location. And this is going to be the germline and the tail of the embryo. Okay, these are three genes, Vasa, Nanos, and Oscar, from this highly conserved genetic toolkit. Well, what about the carpenter ant with the bacteria, with this symbiosis with bacteria? What does its germline look like, especially since it was completely surrounded? Now, in the oocyte, there's just a single location. But as soon as the egg is laid, we discovered something remarkable. And that was that not only is there one location to these germplasm, but there are four. There are actually 4 zones, what we call them, or 4 sets of locations in the embryo for these genes. And we've never seen this in any insect, ever. This is something completely new. And so we were struggling to understand what this meant. Is it possible that this egg had 4 germlines? That's impossible. So we wanted to know what each of these zones or locations of these embryos are doing. And so this is just to show that this is a fresh egg. It's a single cell. It comes right out. The queen lays this. It's a fresh egg. It comes right out. A single cell. They get localized to each part of these 4 zones. And then as the egg starts to divide and cellularize and come up and become composed of thousands of cells and millions of cells, these locations remain. Okay, so the addresses that are set up for these locations early on, they stay later in embryogenesis. And so what we wanted to know is, you can't just have 4 locations of these things that's never been seen. Like I said, the cousin flies, wasps, which are the cousins of ants, or the cousin of the carpenter ant, have 1. You can't just have 4 and not have it affect the way you develop. And so we wanted to know, what is this effect essentially? So this is what the common garden ant, the cousin, looks like. Here is this single germline. In magenta color over here, this is the actual embryo, because most embryos grow in the posterior half of the egg in insects. And this is the tissue. And so we wanted to know, well, what about the carpenter ant? And something amazing again happens. Somehow, not only do you have 4 of these zones of these germline genes, or locations of these germline genes, now you see that the embryo is completely radically reorganized. Instead of now in magenta, the embryo growing in the anterior, it's now in the posterior. It flipped the axes. It went completely to the other side of the egg. It's in the posterior half. These extra embryonic membranes in the anterior part now separate the embryo and the white, which is the Blockmania bacteria, and now sitting in the posterior half. The germline that usually sits at the posterior is now beside the bacteria. And in green, we see a structure we've never seen before in insects, which is essentially, we had to come up with a new name for it. It's called the germline capsule. And it seems to surround this germline, and almost as if it's protecting it from the bacteria. So we wanted to know, what the heck are these different zones doing? And so we knew the germline, these are the bacteriocytes, these are the embryo, this is the extra embryonic membrane. And just to see, I just wanted to zoom in on this part of the bacteria again. At this stage, here again, this is the nucleus, and each of these cells is the bacteria, with the bacteria coming in. They almost look like honey, the honeycombs of honeybees. It's quite remarkable structure that you see it. Well, in the germline capsule, you see a few bacteria that get passed on to the next generation. So most of the bacteria sit here, and these are the ones responsible for immunity, and other amazing functions. And you've got just a few bacteria here that will get passed on to the next generation with these germ cells. And so what we wanted to know is, okay, well, what are these different 4 locations going? Well, we knew from just looking at the egg, following them, that this location, instead we thought that this was going to be the germline. It's not. The germline, this is what's going to form the cells that are going to get passed on from next generation to one generation to the next. This one is actually expressed in the bacteriocytes. Over here, this is the embryo. One of the zones is expressed in the embryo, and the other one is just next to the embryo and curves around. It goes into the bacteriocyte. So what the heck are they doing? Okay, let's look into this. So here, this zone is helping actually form the tail end of the embryo. This one, like I told you, is the new germline. What about this? What's this zone? What is zone 1 and zone 4 doing? Let's take a closer look. Well, zone 1 is right over here, and what it's doing, that it's these germline genes are expressed in the middle of these bacteriocytes, and what we think they're doing is that they're actually attracting the bacteria into the bacteriocytes, helping them get packaged in. Well, what about this zone 4 that's sitting here abutting the things? You can see that it's not in the embryo, it's alongside the embryo. And what it does is it reaches in and it curves into the bacteriocytes. So what we think is happening is it's guiding the bacteriocytes into the gut. And so you can see here, it's something remarkable. I mean, I've never seen this before in any other insect. It's just mind-blowing that you see the, this germline, it almost like extends out, reaches out to the bacteria and says, hey, bacteria, follow me. And they follow it along this path, and that's how they get into the future gut of the embryo. Quite remarkable. Okay, well, one of the things that, that kind of told us, well, this is what these 4 locations are. So zone 2 is where the germline is. This now helps play a role in the bacteriocytes, packaging the bacteria into the bacteriocytes. This one is guiding the bacteria into the future gut. And this one is playing a role in the embryo. So you go from just without the bacteria to having one single location to now having 4 locations. And it's almost like, this bacteria somehow coordinated with the ant, symbiosed with the ant to give these different locations to help the bacteria move into the cells, go into the gut, separate the embryo into the posterior pole, and make a new germline. Just absolutely remarkable. A whole, basically coordinate system to make this happen. Okay, so now as we're going along, we're just going to try and sort of tell you just 3 other parts of the story very quickly. Well, one of the things that we discovered, I mean, this was a discovery that in the bugs where the ancestor of this ant bacteria, the Blockmania, came from, we realized that actually these Hox genes that I was telling you about, that Mateen had tried and failed, are actually expressed in bacteriocytes in the aphids, which are bugs that's where essentially the ancestor bacteria came from. And so we wanted to know, do these Hox genes, which are usually expressed at the very development and tell which, that the antennae should grow here, the legs should grow here. We wanted to know, did these genes play a role in the symbiosis? Now, if we look at the cousin, the one without the bacteria, this cousin looks like any other gene. So I told you, it doesn't get expressed early in the development. These Hox genes come on very late, and they're expressed late, that help, of course, tell the identity. You're going to be the abdomen, you're going to be the head, this is where the legs are going to grow, this is where the wings are going to grow, and so on. But what about with bacteria, the Carpenter ants with the Blockmania bacteria? When you look at this, something amazing is happening. Now, these Hox genes are coming on really early in development, okay? And essentially, you see these Hox genes that are normally never on early in development, you see them in the same 4 zones as the germline genes. And then basically it's expressed later in development, as it always is. And so instead of normally being expressed very late, now 2 Hox genes are actually expressed very early in the very first stages in the same 4 locations as the germline genes are. Okay, and I'm just going to remind you that when you mess with these germline genes, remember I told you these germline genes tell basically where the germ cells are going to form at the posterior, and then it tells it that this is going to be the tail end of the embryo. And if you mess with it, you truncate the embryo. And if you remember Mateen's early experiments, when he tried to knock down UBX, he actually truncation. And so we thought we failed. But in fact, what was happening is when he knocked down, here's again, this is what a normal embryo looks like. When you knock down these Hox genes, again, you're truncating the embryo. You're getting rid of the whole posterior half. And the reason is because when you knock down these genes, you're down regulating all these genes that tell the germline genes, you're basically turning them off. And so basically what Mateen thought was a mistake, and we had given a project, actually turned out that it was because of the bacteria that these genes were actually now expressed early, and actually on top, they were actually regulating these germline genes. And that's why if you knock down UBX or the gene or an ABDA, those genes that we knocked down at the beginning, they actually cut the embryo in half. And so what we thought was a failure and we were about to give up actually turns out to be a major discovery. What it turned out to be is that this whole way we understand the way the plan of an animal is formed, here what it turned out to be is that this whole network was rewired because of the bacteria. And this was a major discovery that we almost threw in the garbage. And this is why, if we didn't find out about Blockman, if we didn't understand it was bacteria, Mateen had never gone to France, if he'd never met up with Junie, all these serendipitous events, if these things never happened, that major discovery would have sat probably in a drawer for the next 50 to 100 years. Okay, so now what's remarkable, look at we return to this egg. And if you look at this, this cloud again, which we now know is the bacteria, these 4 nuclei are the ant. These are the first 4 nuclei of the ant. This is ant DNA right here. This is the bacteria DNA. And at this stage, a single cell stage, this is a single cell. It's a freshly laid egg that comes out of the queen. At this stage, 97.7% of the DNA is the bacterial DNA, and only 3% is the ants. And so we thought, oh, for sure, it's it's the bacteria which is setting up these 4 zones. 97% of the DNA is the bacteria DNA, and only 4%, 3% is the ants. And so we used an established method in the field. Of course, we treated the eggs with antibiotics so we could actually get rid of the bacteria. And when we did that, we thought we would get rid of these 4 zones and turn it back to just one, of having 1 zone. Because if the bacteria is doing it, you get rid of the bacteria, you should go back from having these 4 locations or zones to just having a single one. But when something remarkable happened is that when you do it and you look very early in development, when you treat it with antibiotics, those 4 zones are still there. What does that mean? That means it's actually the ant that is setting up the 4 zones. And so the way you have to think about it, okay, is, it's the ant itself that is asking the bacteria to dance. Okay, it's asking it to dance. It's proposing the dance to the bacteria. It's setting up the 4 zones. And then later in development, much later, the antibiotic treated ones now start to lose the zones. And so the ant then proposes the dance to the bacteria takes over the dance and starts to regulate these 4 locations. And so it's this beautiful cooperation. It's not just the bacteria doing everything. It's this beautiful dance between the ants and the bacteria. Now, my last point, which in about 2 minutes, is how did this whole thing start? These are major changes because of the bacteria, what we call radical alterations. In the cousin, looks like any other insect. The cousin without the bacteria, the common garden ant, looks like any other insect. Now the bacteria in the Florida Carpenter ant now is all of a sudden totally changed. The way where the embryo is, where the bacteria are, where the germ cells, you have new structures. You have all of a sudden, you have 4 locations instead of 1 of these remarkable germline genes. The whole network of how we understand a body plan to be set up is rewired in these guys. And how the heck does that happen? Now, according to the literature, most biologists, going back to Darwin, we think of evolution as happening in small steps, kind of in a stepwise process, very gradual in nature. But in symbioses, all the early theories of symbiosis had these things happening in one major step, saltational step. In the most recent, in a single saltational steps leads to these major symbioses that we've talked about. And so we wanted to know, I guess based on this previous theories, we had predicted that in the ancestor of the, of the Carpenter ant and the bacteria, this whole group that evolved this symbiosis, that we should see all these changes happening in this small steps. And so again, Junie and Mateen did this remarkable work where we looked at more than 30 species of ants to try and reconstruct the step-by-step process by which these 2 unrelated organisms, the bacteria and the ant, came together. And all of these, the origin, I have it here, node 17 here. This is where the obligate endosymbiosis happened. In yellow, here's the Florida Carpenter ant that I just showed you, Camponotus floridanus. All these have the obligate endosymbiosis with bacteria. And so we predicted, like I told you, that basically at this node, that we would see all the changes appearing in this node. Something remarkable happened. And we started looking, this is the cousin, the common garden ant that I showed you over here, without the bacteria. Well, what we see in these, in the common garden ant, of course, it looks like any other insect. The embryo grows in the posterior and it has a single location. But here's the big surprise. In all these ones with gray, this is what the embryo looks like. It looks like any other insect. But in these nodes, just leading up to this symbiosis, in 14, 15, and 16, something remarkable happened. Now we see, instead of just one location, we see two locations. Almost as if but the embryo is still in the posterior half of the egg. It's almost what we call this a pre-existing capacity. It was almost a change that was necessary for this symbiosis to happen. When we looked at the origin, we thought we would find at least 4 zones. We only found 3. So in these early branches, they only had 3 zones. It's only in these light green ones at the top that now we get zones. And so the way this happened, instead of just like this single saltational step that happens in one fell swoop, now all of a sudden you go from 1 location to 2 locations. And this 2 locations then help set up, it's a pre-existing change that helps set up, the bacteria comes in, it takes control. Now you get 3 locations, then finally you get 4. And so we think that these zones, instead of happening in one saltational step, happened in a step-like, gradual step-like manner. And so all these amazing symbiosis that I gave you examples of in the tree of life, whether it's algae and bacteria and humans, how bacteria and humans came together, we think these are the things that are necessary for that to happen. We think that there has to be rewiring of networks. We think that there has to be pre-existing changes that help these symbiosis to come together. We think that there has to be some ecological interactions, like what I talked about, the bug and the ant. Basically the ant drinking the butt juice. That pre-existing ecological interaction, together with this pre-existing change, together with the rewiring of the networks, these are all the kind of the secrets of symbiosis for symbiosis to actually happen. And with that, I would like to say that, again, symbioses are the rule and not the exception in life. And I think we've made a major discovery towards trying to understand the steps towards how these things happen. And I would just like to thank all the funders for this work and all the supports. And again, just like to thank the students, Junie and Mateen, and all those in this who joined me in this serendipitous journey, from Lloyd Davis and others who helped us collect the ants. It's really a big team effort that helped get us this far. And this work, of course, was published in Nature magazine, and it made a big splash when it came out. You can check out the McGill news pages back in 2020. Thank you so much.
Thanks, Ehab. That is quite a story, and we do have time for questions because we had a little delay at the beginning. Adeline, Anouk, please moderate our question period.
Yes, that was fascinating. Thank you, Professor Abouheif. And we do have a lot of fascinating questions. So the first one is 51 million years ago. It was the start of the what was happening at that time? Well, it was the start of the mammals after the dinosaurs went extinct, right? So would you say that is significant in terms of the story you just told? Like, were ants around while the dinosaurs walked the earth And could we say that there has to be some primitive ants and bacteria, and could there be any relationship between them?
You know, I don't know of any direct relationship, but life is all connected. All these events are connected. I'm not a paleontologist, but any paleontologist would tell you that during these events there are millions of things happening at the same time, thousands of events happening. So how they're all connected are things that we're just starting to understand, but we have to understand. So that, I don't know of a connection, but it doesn't mean there wasn't one. There's all kinds of amazing connections. And of course, we owe a great debt to the flowering plants, because without the flowering plants, there's all kinds of symbioses that happen because of the flowering plants and insects. It was a love affair between the flowering plants and the insects that came along during the early history of life. And who knows if there was connections between the dinosaurs. And I mean, the history of life is full of fascinating connections. And my colleague Hans Larsen has spent his whole life trying to figure that out.
Yep, fascinating. Thank you so much.
And we have another question for you. So is the type of bacteria that gets passed on to the next generation through the capsule in the Carpenter ant, is the type of bacteria always the same?
Yes. So it's, like I said, it's an obligate relationship. They can't live without each other. And so what's even remarkable is that when you look at the different, like the species of Carpenter ant, if you compare the tree of the bacteria and the tree of the Carpenter ant, they mirror each other. So when the Carpenter ants were speciating, the bacteria were speciating along with them. And so it's really, again, a remarkably tight relationship. They're almost behaving as an individual, and so the bacteria is the same one and goes along. Now, the human body, the association, there's other bacteria that are around in the ant, but this one has a special relationship with the Carpenter ants. Yeah.
Are there any more questions?
Well, if we have time.
Yes, one more.
Perfect. So just going back to the very beginning of the talk, could you explain a bit more about the Evo-Devo? Like, what do you think of it?
So I'll tell you a funny story. When I was, because I had been awarded the Guggenheim, I had the honor of meeting Mona Nimmer, which is the President Trudeau's advisor of science, scientific advisor. And I told her, she went around the room asking what people were studying, young scholars who had won recent awards. And I said, well, I study Evo-Devo. And she said to me, oh, that's interesting. She laughed and she said, oh, that sounds like something I would expect to find on the menu of Air Canada, an Air Canada flight. And I just looked at her and I said, well, in fact, I think it's going to be the future of biology. And so what I think of it is, I do think the future of biology is going to be Evo-Devo, because although we are remarkably focused on genes in our societies and what genes are doing and the causal effect of genes, genes are not the primary cause. They are part of the equation, and they interact in very amazing ways with their environment. And so everything from our behavior to our development to our evolution to everything is extreme. This gene environment interaction is absolutely tied. And so this field, I think we're just starting to realize that everything we know about molecular mechanism is all the species that we study are called model organisms. There's about 10 species that all of medicine and molecular biology are based on. And we ignore the more than 2 million described species on the earth. And what Evo-Devo is saying is, guys, we have to look at all of these species. We have to look at all of these genes. We have to look at all their environments, because as you can see, what I hope I've shown to you today, it's the interaction of genes with their remarkably rich environments. It's putting these genes in their real ecological context. in symbioses, in all kinds of things that are going to determine the way we look, the way we behave, the way we evolve, and many other things. It is the future of biology.
Well, I think we've gone on quite a journey. Molecular biology has never been more beautiful. Those images and we're gonna the world of eco, evo, and devo has never been more beautifully presented. So I officially am thanking you with a few gifts. Ehab, this is a bookmark. Can you see the signature there?
That's so exciting.
So we have a letter written by Charles Darwin. It is at the Red Path Museum, and we've extracted a bit of that letter and made a bookmark for you.
Wow.
We hope that that bookmark will be part of your reading. The Fossil's Tale is a book we published at the museum, the Red Path Museum at McGill, about all the history of life based on the exhibits in the gallery. So the museum's not open, but you get to go and visit the museum by looking at this book. It's basically a gallery guide. Okay, so your bookmark from Darwin goes into your Fossil's Tale. If you have to do some outreach or go shopping, please wear our outreach face mask. I'll be sending some inserts as well with that, some filters.
Great.
And then if you just have nothing better to do and just feel like having fun with a stereoscope, we had a photographer come to the museum. He did a stereoscopic photo, and this will pinch open and you get to see the dino skeleton in 3D.
Oh boy, you know who's gonna love that? My 5-year-old son, Adam. Oh boy,
Good.
he's gonna love that.
I'm glad you can share your things with your
Yeah.
family, and please with your students too. Definitely keep this in the lab. I think it'll be a welcome addition.
Absolutely.
Thank you to not only our speaker, our ambassadors, Adeline Lee and Anouk Arsenault, students at McGill University who've helped us moderate, who were introducing Ehab, and we will be able to continue this series monthly, every Thursday night. Next month, February 10th, same channel, YouTube channel, 6 o'clock. It'll be a speaker from the School of Occupational and Physical Therapy. So we're leaving the molecular world and we're going into the world where people actually study human beings and how they respond, how they're conscious of their movements, the way their muscles move, and how their brain regulates some of those conscious movements. Her name is Stephanie Blain-Moret. See you back here in about a month's time, February 10th. Thanks again, everyone.